The overall goal of this procedure is to determine ultrasonic vocalization, or USV preferences in mice using a two-choice playback test. This is accomplished by first recording USVs from a live individual. The second step is to edit and trim recorded USVs accordingly and generate stereophonic sound files. Next, the ultrasound emitters are calibrated and the USV volume is adjusted using the attenuator and amplifiers. The final step is to introduce the subject mouse into the test box and start playing the two sound files simultaneously. Ultimately, behavioral monitoring can reveal if the mouse shows a preference for one sound over the other.
The USV emitters we have developed can help us to answer key questions concerning mice vocal communications, such as what is the biological significance of different types of USVs.
The main advantage of our acoustic emitter over existing devices like piezoelectric transducers is that the frequency response has no resonant peaks. The broadband flat emissivity in the ultrasound vision with no harmonic distortions makes it possible to use the emitter for reproducing complicated acoustic communication calls.
Through this method can provide insight into the female response towards USVs of males. It can also be applied to other type of social interaction, such as between mother and pups, between adult males, and between adult females.